All right, so thanks everyone for coming. I'm Lisa Komoroske. I'm at UMass Amherst. And I work mostly in ocean ecosystems, where a lot of our work is broadly thinking about how evolution shapes marine ecosystems, particularly under rapid environmental change. And as our thought in the field about how this works has changed in past decades, I like this quote. I think Resnick et al. Put it nicely, saying evolution was formerly thought of as a historical process perceived through the footprints left in the fossil record or living organisms, but now it's seen as a contemporary process that can act in real time. And I think a great example of this is our understanding of local adaptation in the sea. So if we think about the historical paradigm, where we've got larval stages of these beautiful larval fishes and invertebrates here that are in the water column and can travel long distance and go through the water with few physical barriers, that this should promote high genetic connectivity and everything should be homogeneous. But now we can appreciate that actually there are really strong gradients and mosaics in our oceans, and also really interesting physical and behavioral retention mechanisms that can promote local adaptation. And so we're now seeing and appreciating that it's more common than we previously realized. And in fact, a meta-analysis last year found that local adaptation in the ocean may even be more common than in terrestrial systems. Now, when it comes to climate change forecasts and vulnerability assessments, largely in the ocean, we're still thinking or assessing them based on, everybody's the same. And while we're understanding that there is more local adaptation existing, there's still a lot of things we don't understand about it. And so I will argue that studying these microevolutionary processes can give us really important insight into how this variation is generated, maintained, and tell us also about potential future adaptive capacity. So I put up this, simple figure that I think a lot of us have in our talks of, okay, ultimately we're trying to put together the pieces of, how the environment is linking to a phenotype and the underlying genetic mechanisms. And it looks really simple, but we all know it's anything but simple to figure this out. And a lot of the approaches that we have, especially in natural systems that are very messy, have the strengths and weaknesses. And so one project and approach that we're trying to take to kind of put these pieces together is an integrative approach. So along with my fellow snail enthusiast, Brian Chang, and a great team of postdocs and students and technicians, we've been working with a marine snail, Urosalpinx cinerea, to connect field collections and surveys with manipulative lab experiments, so long-term pseudo-reciprocal transplants and common garden experiments in different environments, and then also molecular analysis. So today I'm going to focus just on a couple questions relating to the molecular analyses, but if anyone wants to talk about the other parts, what it's like to try to raise up thousands of baby snails in the lab for years at a time, come chat with me. And so ultimately, what I'm going to focus on are two key research aims. So I'm going to talk about how we quantify population connectivity in the system and use it to reconstruct invasion history, and then put that in the context and put our evaluations of genomic signatures in that context to understand potential environmental adaptation. So spatially, thinking about local adaptation across their native range, as well as potentially temporally, because in this context we have some invasive populations that we can make predictions and then test those predictions. So, okay, let's get to know Urosalpinx, the Atlantic oyster gill. Don't be fooled by this adorable picture. It is a voracious predator. So they are called oyster drills because they literally drill through the shell of bivalves with their radula and chemical secretion. So here is an oyster that attempted to be eaten 4 different times. Luckily, it persevered. But they're a great study system also because they are direct developers. So in the lab, they lay these egg capsules that are full of tiny baby snails that we can put into experiments and account for parentage and other things like that. They're also seasonal, they have seasonal reproduction, and they're native in the Western Atlantic and North America. But about 100 years ago, we thought it was a good idea to put thousands, or actually millions of oysters on railroad cars and ship them over to the west coast of the US, and they brought along their friends. So Urosalpinx now spans thermal gradients on 2 different coasts. And so on the east coast, they range from down in Florida up to Maine, maybe even in Canada, and this is one of the strongest thermal gradients that exists in the world. And then over on the west coast, we have the invasive range from mid-California up to Canada. And if we look at some representative environmental data across these ranges, you can see that the temperature profiles over the course of the year vary both within and between these ranges. And so if we're thinking about local adaptation, we might first ask what could be the important environmental drivers. And so first work that we did was a master student of Brian's, Drew Villeneuve, who did some nice thermal performance curves looking at growth and maximum trait performance. And found a really nice relationship between maximum growth and season length, and this is in the Atlantic and the Pacific. So evidence that there's local adaptation and growth that's correlated with season length. But we might ask, well, okay, why don't all populations grow quickly? We might think that there are potential trade-offs. And this becomes important when we think about potential future projections, right? So if we think that the northern sites are going to extend their warm season, do we expect that we'll see reduced growth rates? And I think it really depends upon a lot of different factors, including what the genomic architecture is of the genes under selection, if there are indeed physiological trade-offs, and as well as potential constraints through population history and genetic connectivity. And so we sought out to kind of tackle some of those questions. So we went out and we collected snails from across their native range as well as their invasive range. And this is work led by my former postdoc, Blair Bentley. And we generated genome-wide SNPs using RADseq. So first, if we just look in the native range, going from, I have a little pointer, but go from left, your left to right. From Maine down to Skidaway, Georgia, we see really strong regional structuring. And this is reflected in the DAPC as well, where we see that as well as some substructuring within regions. And these fall into a nice IBD pattern. I have color-coded the contrast by within and between clusters. I think it's a little too small to read. But basically what we see, what I want to point out, is that there's a group that's kind of below and a little bit above. And those are likely due to kind of classic biogeographic breaks at Cape Hatteras and Cape Cod. And so overall, we can now have a good idea of the connectivity on the native range. And this makes sense, right? So they're direct developers. The snails hatch out. They can only crawl so far. Maybe they can hop on a kelp frond and draft somewhere, but not surprising. So now we can turn our attention to, okay, they're existing along this really strong gradient. Do we see evidence for local thermal adaptation? And so we can use NOAA data with their temperature traces for all of our sites and extract some metrics for different thermal variables that we might be interested in and thinking that might pose selection pressures. And so if we do this, and then we put them into, we use redundancy analyses to look at the relationships with allele frequencies and our environmental variables after accounting for our population structure, so in the partial RDA. We identify a little over 200 putatively thermally adaptive loci, and again, this is just in the native range. And interestingly, most of these loci have strongest links to annual mean and max temperatures. Okay, so what's happening in the invasive range? So over here on the west coast, to bring up our native structure plots again, what I didn't show you before is that actually all of our invasive populations and individuals fall into that bottom left cluster, so they're hiding behind the other points. And when we do actual assignment analyses, it's a little hard to see the colors, but basically all of the invasive, the individuals from all invasive populations assigned to one of our southern New England populations, so either in Massachusetts or Connecticut. There's one weirdo from Delaware Bay. I don't know what's going on with that. But basically this lines up with what we know about the invasive history, where oysters were brought up to Long Island, to southern New England, and then moved cross country. So now what we can do is think about what environment they came from, right? And then how that differs from the environments that they got moved into, and think about questions of local adaptation in their new environment. And so conceptually what this entails is thinking about, we've built these relationships for every locus where we have, I wish I had a little pointer, but if you look on that line, we've got all of our circles are our points in the native range, and our source populations are the red and the blue dots. And if we saw a signature for adaptation at our putative adaptive loci in the invasive population, we would expect that they would move to stay on that regression line, right? and that would be suggestive of adaptation to that new environment. And so what this might look like on the right over here is that when we look at differences between the source and the invasive populations, depending on where you start, they might, the pink and the green, you might end up in different places. But the really important part is this yellow is the difference that we'd expect to see in the predicted allele frequencies with the actual allele frequencies. And if we expect them to be on that line, they should hover around 0. Now, alternatively, we might see the opposite pattern, right? Where the allele frequencies in the invasive populations are more driven by the source population, what they were before, and this might be caused by relaxed selection or some other population constraints. And so what we would expect to see in that case is actually a reversal where now our yellow violin plots are differences in the regression or differences between the invasive and the regression line are off that 0. So if anyone's interested in the nitty-gritty details of how we did this, come talk to me. I don't have enough time to go over it, but basically we do this iteratively for all of the loci, and then we also randomly sample neutral SNPs to make sure that any divergence that we're seeing is not just drift or random variation. And overall, what we see is this situation where there's not clear evidence for rapid adaptation, at least at the loci that we predicted were putatively adaptive in the native range. That could be indicative of relaxed selection. Now, I think what's really interesting about this story, well, okay, I think everything related to these snails is interesting, but Eurosalpinx is a really prolific invader. So a lot of times we think about, genomic vulnerability and projected shifts in allele frequencies, and the idea is that if they can't do that, then they have the likelihood of persistence, right? We didn't see evidence of that here, but they are doing just fine. So this is actually a photo of a community outreach event that Brian organized with Audubon, where they eradicated, I think, 30 to 50,000 Eurosalpinx. Did not make a lick of a difference. It's really hard to eradicate invasive species. And so they are able to proliferate in these new environments without adaptation at these loci. Now, the last thing I just want to talk about really quickly is, what else could be going on. So this is some thermal tolerance work done by another postdoc in the lab, Chris Swain. And what I want to just point out is that these two lines, so the dotted line are snails that are acclimated to 22 degrees Celsius, and then the lower line is 12 degrees. And what should jump out at you is there's not a lot of difference in thermal tolerance across populations, but there's a lot of plasticity. And so it could be that plasticity or the capacity for plasticity is playing an important role in their ability to persist. So some key takeaways. We see evidence of local adaptation at both phenotypic and genetic levels, but we're still trying to piece together how it all fits together. But that hopefully I've shown you that by coupling invasion reconstruction using population genomics methods with environmental data can be informative for understanding local adaptation as well as making and testing predictions. We see that Eurosalpinx has successfully established without clear evidence of rapid adaptation. This could be due to relaxed selection and potential high plasticity, or of course there could also be adaptation happening in other loci that we didn't investigate or other traits that we need to look into. So if we think back to our simple schematic, I think we've made some progress, but we still have a lot of things to figure out. So some ongoing work that we're doing, I mentioned the thermoplasticity work. Another postdoc, Allison, is looking at trade-offs with reproductive output and energetic allocation. I have a new postdoc, Sneha, who's looking at some of the differences from our experiments in phenotypic traits and looking at the genes that might be related to that. I'm going to cut off. And I have a student looking at transitional plasticity. So hopefully next year I'll be able to update you. Okay, well that wasn't actually my gong. Okay, well, I have 30 seconds left, so I will acknowledge lots of people, all my co-authors, funders, and people who helped us collect and take care of baby snails and sample them. So I think I have a short time for questions, and I can also talk to people after. Thank you.
All right. Thanks everybody for coming. My name is Robin Waterman, and I'm a fifth-year PhD student at Michigan State's Kellogg Biological Station working in the Connor Lab. And today I'm going to be talking to you about some of my work on evolution and plasticity in a native agricultural weed. So I'm really interested in this group of plants we call agricultural weeds. And these are just plants growing where they aren't wanted, that compete with the cultivated plants that we do want to grow. And I think that studying agricultural weeds provides immense opportunity to advance our basic understanding of how plants colonize novel environments, particularly given that agroecosystems are now a major global habitat type, comprising almost half of Earth's habitable land surface. We also know that weeds can seriously reduce the yield of all our major crops when they aren't controlled. The problem is that current control methods are often unsustainable and expensive. So my hope is that a more thorough understanding of how plants become problematic weeds will help us design more successful and sustainable management strategies for the future. So I'm going to talk to you about a few hypotheses for how agricultural weeds colonize these agricultural habitats. So in this conceptual diagram, let's imagine that this blue dot here is a non-agricultural population expressing a trait value at the optimum for its local habitat. Now, in order to succeed in the agricultural environment, it needs to, in some way, get to increase its trait value and get to the new optimum shown in that red star. One way this can happen is through adaptive plasticity. So in this scenario, the population is what we might call pre-adapted to invade cropland. No evolution is needed since the population already expresses the optimum trait value in the novel environment. The second option is that the population invading the crop field may rapidly evolve toward the agricultural optimum, resulting in a pattern of local adaptation. Third, non-agricultural plants that invade an agricultural environment may initially express plasticity in the adaptive direction, but that is insufficient. But this might allow time for the population to persist and for selection to further move the trait mean toward the optimum. To test these hypotheses, I used the species common ragweed. It's a weed that's native to North America with populations found in agricultural and non-agricultural habitats. It's monoecious, so it has separate male and female flowers on the same plant. And in this picture, you can see these, or you may be able to see, these spike-like clusters of male flowers, and they produce giant amounts of wind-pollinated, self-incompatible pollen, but you may already know that if you suffer from fall allergies. I'm sorry. So my first research question is, are there plastic trait differences between ag and non-ag environments and/or evolved trait differences between ag and non-ag populations? And my second question is, whether any such evolved or plastic differences were adaptive. So in year one of this experiment, I collected seeds from 20 to 30 plants from paired agricultural and non-agricultural sites. I then grew one progeny from each field-collected parent in a common greenhouse environment to standardize parental effects and generate families of seeds via random within-population mating to use in subsequent field experiments. And then what I'll be showing you today is results from year 3, in which I reciprocally transplanted the seeds sourced from both non-agricultural and agricultural habitats back into both a non-agricultural old field habitat and an agricultural organic soybean field environment at the Kellogg Biological Station in 2 replicate spatial blocks. So you'll notice in this project I am focusing on organic fields because my goal is to learn more about traits that are important for initial weed establishment before they become targets of intensive herbicide management. So this design included a total of 1,080 plants, and my sample size at harvest was 397. So starting off with this first question, and in the interest of time, I'll be focusing on the 3 most interesting traits for this talk, but I'll note in a few cases when there are other traits that were qualitatively similar. I'm going to be showing several plots like this with the growth environment on the x-axis, non-agricultural is on the left, agricultural is on the right, and then along the y-axis is the estimated trait value after accounting for source region and block. And then the blue points are means for plants sourced from non-agricultural environments, and the red points are means for plants sourced from agricultural environments. The lines connecting them are their reaction norms. So as we can see from this plot, there was a strong effect of growth environment indicating a plastic response in which plants grown in the agricultural environment emerged faster. However, there was not a significant source environment effect which would have indicated evolution. And the flowering time results mirrored this result, but bigger error bars, so they were not significant. Next, we're looking at stem length. And from this plot, we can again see a strong plastic response to the agricultural environment, causing increased final stem length. And height at first flower was also strongly correlated with stem length and mirrored this pattern. Similar to stem length, plants also had longer final root length in the agricultural environment, and the number of leaves was correlated with root length as well and mirrored this result. So to summarize these findings addressing my first question, consistent with hypothesis 1, I found strong evidence for plasticity in all traits, with plants grown in the agricultural environment emerging faster, being taller when they flowered, making longer roots, longer stems, and producing more leaves. In contrast, I found no support for the second hypothesis since there were no significant evolved differences. Given this lack of evolution, we wouldn't really expect to find local adaptation, but I did look for it anyway, just in case there were any important unmeasured traits that could be driving adaptation. However, as you can see from this plot of total fitness measured as number of seeds produced, the ag plants grown in the ag environment actually had lower fitness than the non-ag plants, and the two source populations did not significantly differ when they were grown in the non-agricultural environment. And third, even if the mean differences between the ag and non-ag populations were significant, the direction of change is all opposite that of plasticity, in contrast with our expectation from the buying time hypothesis. So no evidence for hypothesis 3 as well. In my second question, I asked whether the plasticity that I observed was adaptive. And to look at this, I did selection gradients in the two different environments. Now, the two most straightforward ways to find evidence that plasticity is adaptive is first when selection is in different directions in the two environments. So I'm showing that with the blue downward arrow and the red upward arrow in the direction of plasticity, which I'm showing in the black arrow. Second straightforward way to find evidence for adaptive plasticity is when there's stabilizing selection in both environments, but at different optima in the direction of plasticity. But as you'll see in a moment, there are a range of other combinations of plasticity and selection that are possible and that I'm going to discuss. So let's start with emergence time. So what I'm showing here are standardized selection gradients, so the strength of selection acting on a trait after accounting for selection on correlated traits separately in the two environments. And for this trait, selection acted through the fitness component of survival to reproduction. As shown in this diagram here, the plastic response in which the ag plants emerged faster was beneficial for plants in the ag environment because there was stabilizing selection on the mean expressed in this environment. However, there was not stabilizing selection for later emergence in the non-ag environment as we might have expected, but rather directional selection toward the agricultural optimum. And we found a similar pattern for flowering time as well, but less significant. For stem and root length, the selection gradients I'm going to show you are for female fecundity, so the number of seeds produced by surviving plants. And in this case of stem length, there is opposing selection in the two environments for longer stems in non-ag and shorter stems in ag. But this opposed rather than followed the direction of plasticity, in which plants grown in the ag environment had longer stems. So this suggests non-adaptive plasticity for this trait and for height at flowering as well. And finally, for root length, there was positive directional selection with curvature in both environments, though the linear and nonlinear components were both stronger in the agricultural environment. Similar to the case for emergence time, plasticity to increase root length in the agricultural environment was beneficial for plants growing in the agricultural environment, but shorter root length in the non-agricultural environment was not beneficial to these plants. So to sum up what I'm finding in this project so far, I first found strong evidence for plasticity. Plants in the agricultural environment emerged faster, taller at flowering, had longer stems and roots, and produced more leaves. But no traits showed significant evolved differences between ag and non-ag populations, and these populations were not locally adapted. So overall, this is consistent with a scenario of very high gene flow across environments. And this might be one way in which the invasion of local weeds into nearby crop fields may differ from invasions across continents for our sort of classic invasive species scenario. That non-agricultural source population may often be quite close by and providing continual gene flow into agricultural habitats. Second, I asked whether such differences were adaptive, and I found that emerging faster and growing longer roots was adaptive for plants in the ag environment, but the opposite response was not adaptive in the non-ag environment. Although selected for in both environments, it may be particularly beneficial for weeds in ag environments to emerge early, so they're big enough to survive early season mechanical weed control, and similarly to grow longer roots for better anchoring against being uprooted by that weed control. In contrast, growing longer stems in the ag environment was not adaptive. The plastic response here seems to have overshot the optimum, and this may indicate that plants invested too much in growth and not enough in reproduction, given that time constraint of being killed at the soy harvest. So in all these cases, the reaction norms are not optimally adaptive, and the reasons for that this has not evolved are a bit unclear. They might include correlations with unmeasured traits, fluctuating selection over time, or opposing selection through male fitness. And that's something I'll be looking at more in the future. So next steps are to process remaining data, and I'm conducting a second temporal block this summer with some improvements. And I'd just like to thank everyone that helped me in the field and provided feedback on the project. Thank you.
So I'm Elizabeth Makovec. I'm a first-year PhD student at the University of Toronto, and today I will be discussing the role of sexual selection in immigration and adaptation to a novel environment in Drosophila suzuki'i. So in thinking about sexual selection and adaptation, theory focuses on the alignment of sexual and natural selection. Theory predicts that when sexual selection opposes natural selection, this will not promote adaptation. Specifically, it suggests that traits that are good for mating success or other sexually selected traits may reduce fitness in other ways. However, sexual selection may align with natural selection. Under the good genes model, it suggests that sexually selected traits such as display traits reflect genetic quality. And so these traits, when they're sexually selected, will then promote fitness because they reflect genetic quality. Furthermore, theory has suggested that sexual selection may aid in the purging of deleterious alleles, which aligns with natural selection, again promoting adaptation. When looking at sexual selection and adaptation in lab experiments, there has been mixed results. Illustrated here is one of many results. In this result, sexual selection does not promote adaptation. As we can see here, treatments with sexual selection had no increase in fitness relative to treatments without sexual selection. However, other results, other studies have suggested that sexual selection is associated with increased viability, therefore associated with promoting adaptation. Some research has suggested that reasons for these mixed results is the physical mating environment in which these flies are, in which these experiments are run, may be influencing the alignment of natural and sexual selection, and therefore whether or not sexual selection promotes adaptation. So when thinking about sexual selection occurring in complex mixed environments, illustrated here, it has been found that sexual selection may promote adaptation to novel larval environments. As illustrated here, we can see that sexual selection, specifically mate competition, occurring in complex environments, has higher fitness relative to sexual selection occurring in simple environments or when there's no sexual selection at all, suggesting that, again, sexual selection occurring in more complex environments may promote adaptation. And so our goal is to continue studying the role of sexual selection and adaptation in these more complex mating environments, but also to investigate the role of immigration alone and in concert with sexual selection in this adaptation. So we're focusing specifically on pulsed immigration. Pulsed immigration is immigration that occurs with generations in between immigration events. And modeling work has suggested that pulsed immigration may promote local adaptation. This is illustrated here where we can see that more pulse forms of immigration are associated with a higher allele frequency of a beneficial allele in a sink environment or an environment in which outside an organism's normal niche relative to more continuous forms of immigration. And this is suggested to be because pulse immigration offers repeated opportunities for genetic rescue, but also reduces maladaptive gene flow. So I'm looking at immigration and sexual selection in relation to adaptation in Drosophilus suzuki. Drosophilus suzuki is an invasive pest species that originated in Southeast Asia and has since spread throughout the world to diverse environments. It's also a crop pest species, and looking at sexual selection and immigration in relation to adaptation in this species may shed light on how the species is able to rapidly adapt. Furthermore, when looking at Drosophilus suzuki and adaptation, not only has Drosophilus suzuki been shown to adapt to multiple different fruits. As illustrated here, we can see that adaptations to one fruit may be adaptive to other fruits, as illustrated by how increased fitness in one fruit environment was associated with increased fitness in another fruit environment, suggesting that populations that are adapting to different fruit environments may have adaptive alleles if they were to immigrate between these fruit environments. So again, I'm investigating the role of sexual selection and immigration in the adaptation to a novel environment in Drosophilus suzuki, specifically looking at how sexual selection and immigration alone or in concert affect adaptation and how this manifests in terms of fitness. We predict that immigration and increased opportunity for sexual selection will increase rate of adaptation. So for this, we are conducting an experimental evolution experiment in which we introduced 12 populations of Drosophilus suzuki to a novel food, this food being raspberry food, and allowed them to evolve. Currently, the populations are on generation 30 on this food. We have 4 treatments and 3 replicate populations per treatment. For each treatment, we manipulate opportunity for sexual selection and immigration. For manipulating sexual selection, we have our plus and minus sexual selection treatments, where plus sexual selection, there's increased opportunity for sexual selection due to the complex mating arena as well as multiple mates being in the arena. Whereas our minus sexual selection treatment, the flies are undergoing enforced monogamy, so there's less opportunity for sexual selection. And then for immigration, we are manipulating immigration by introducing immigrants from a strawberry population of Drosophilus suzuki to the respective mating arenas. And in terms of adaptation, we're looking at adaptation in terms of fitness. Specifically, we're looking at fecundity, or the number of eggs laid, and reproductive fitness, or the total number of adult offspring. So for our fecundity assay, we take flies from our experimental evolution populations and introduce them to a egg-laying bottle and allow them to lay eggs for 24 hours. And then after 24 hours, we count the eggs. This plot here represents the average number of eggs laid by 25 females in 24 hours per treatment. Along the x-axis, we have the generation sampled, and then along the y-axis, we have the number of eggs. Here, the blue colors refer to populations undergoing increased opportunity for sexual selection, and the reddish colors refer to populations that are undergoing no sexual selection. Light colors refer to no immigration, and darker colors refer to populations that do have immigration. And so from this plot, we can see that overall, all the populations have increased in fecundity over time, suggesting that all of these populations are adapting to their novel environment, at least in terms of fecundity. And we also see some treatment differences. At generation 26, we can see that populations undergoing increased opportunity for sexual selection have higher fecundity than populations that had no sexual selection, suggesting that sexual selection may promote increased fecundity. For our fitness assay, we followed a similar setup where we took a subset of flies from our experimental evolution population and introduced them to our fitness assay setup, and then we collected all offspring emerged over all days of emergence and counted the number of offspring. So this plot, again, represents the average number of adult offspring produced by 3 females and 3 males. Along the x-axis, again, we have the generation sampled, and along the y, we have the number of offspring. Here we can see that while most populations have increased in number of offspring over time, not all have, suggesting that these populations may still be in the process of adapting to the novel food, at least in terms of reproductive fitness. And we also see very few treatment differences between our treatments, suggesting that neither immigration or sexual selection increase reproductive fitness. However, when we look at individual replicate populations, we do see some individual populations are quite different from others of the same treatment, suggesting which may be driving some of the averages that we see. And so overall, we see that potential adaptation in respects to some components of fitness, but not others. Specifically, we see that sexual selection may promote increased fecundity. However, neither immigration or sexual selection seem to increase reproductive fitness. And so there's this misalignment between fecundity and reproductive fitness because our sexual selection is increasing fecundity. However, this does not translate to an increase in adult offspring, suggesting that there's something going on during larval development or between the egg and adult stage. That something's going on in between that stage. And so our next steps are to continue looking at fecundity and reproductive fitness, but also look at how sexual selection and or immigration influence larval development, specifically either looking at number of eggs fertilized or number of larvae hatched. I would like to thank the Stepanek Lab and all my undergrads that helped with this research, as well as my funding source. Thank you. And I think we have time for questions, so. Yes?
So, I know that in Drosophila melanogaster, there's lots of evidence for males harassing
Not that I'm aware of. Based on what I've seen, they're just interacting with the flies. There are instances of male harassment. It is something that I want to look into further. Thank you.